Hello, everyone. Thank you all for joining us this afternoon. So let's wait for 1 more minute for people to join the webinar. And during the webinar presentation, if you have any question, you can post it at the Q&A, and our speaker will answer the question at the end of his presentation. Okay, so now it's 3:01. Let's get started. I see we already have 53 people join us, but we do have around 200 registrations. So hopefully they can join us later. So hello, everyone. Thank you all again for joining us this afternoon for our webinar. I'm Qin Yang, an associate professor at Duke School of Nursing, and also the co-chair of the ICSA Outreach and Engagement Committee. For those who are joining us for the first time, ICSA has recently initiated a webinar series titled Career Development in the Pharmaceutical Industry. So this series aims to bridge the gap between the academic training and the practical knowledge required in the pharmaceutical industry. So Deli, can you help to move to the next slide? Thank you. Yeah, so in this webinar series, we focus on two main themes. One is in soft skill and leadership development, and the other one is knowledge-based training. And for the soft skill and leadership development, last month we had our very first webinar, which is really targeted on the fresh graduate students or junior statisticians. If you missed the webinar, the recording is available at our ICSA website. And so today's webinar actually is the very first one for the theme of the knowledge-based training. The webinar title is Daily Work Life as a Statistician in Pharmaceutical Industry. And the speaker of our webinar today is Dr. Deli Wang. Dr. Deli Wang is the Director of Statistics and Research Fellow at AbbVie Incorporation. Dr. Wang obtained his PhD in biostatistics in 2004 from University of Iowa. He is currently the statistical lead in new drug development for neurodegenerative diseases. He has extensive experience in designing and conducting phase 2 and 3 clinical trials in multiple therapeutical areas, such as neurology, psychiatry, oncology, nephrology, and immunology. He has contributed significantly to statistical methodology development in clinical trial design, particularly in interim go or no-go decision-making, multiplicity adjustment, and demonstrating disease modification in neurodegenerative disease trials. Dr. Wang has published over 70 peer-reviewed paper and book chapters, including 41 of them in the past 13 years, after he entering the biopharmaceutical industry. So without further ado, let's welcome Dr. Wang for today's webinar.
Okay, thank you very much, Qin, for a nice introduction. So let me start my presentation here. So title is Qin mentioned that is Daily Work Life as a Statistician in Pharmaceutical Industry. So this is my disclaimer. I just want everybody to be aware. What I present here represents my personal views and opinion. It does not represent my company's view or position. So in this webinar, So main objective is to provide some background information of daily work life as a statistician in biopharmaceutical industry. So the target audience are students in statistics, biostatistics, or relevant areas, or junior statisticians who have interest in working in biopharmaceutical industry. We try to help junior statisticians in their career choice when the time comes. So these slides show the overall outline of my talk. First of all is biopharmaceutical statistician daily work life. That's our main focus of this presentation. Because relevant to the biopharma statistical daily work life, there are some concepts in clinical trials we are talked every day in the daily work life. So next, third one, I'll just talk about clinical trials reporting, CDX, STTM, item data standards, and the table listing figures. And lastly, I'll give a very summary of final remarks. So for biopharma statistician is a statistician. So we work in a biopharma industry. We share the common work life as a statistician in other fields. For example, we're dealing with numbers, formula, and interact with his or her customers for the projects. But there are some uniqueness of daily work life for a biopharma statistician. For example, pharma statistician provides statistical support in drug development cycles. And we interact with cross-functional stakeholders in drug development process. Those stakeholders are listed here, for example, such programmers, data scientists, physicians, trial operational lead personnel, regulatory personnel, epidemiologists, safety and pharmacovigilance personnel, medical affairs personnel, commercial personnel, project managers, drug discovery or preclinical researchers, regulatory agencies, for example, FDA, EMA, PMDA, and others. So those are very important stakeholders we work together for drug development. So in our daily work life, it involves both operational and scientific works in clinical research. Here I just use a cartoon example to show. Move a little bit. Sorry about that. To show the some daily work life as a biopharma statistician in a cross-functional team meeting. So in this example, the main stakeholders are statisticians, medical director, trial operational lead, and the regulatory lead. So here we move on. So you can see that in the table there's a physician, statistician, regulatory lead, operational lead. So here the physician typically lead clinical trial development. So like I said, I use an example for Alzheimer's disease. She may say, we are developing a drug for Alzheimer's disease, include phase 1, phase 2, and phase 3 trials. And the statistician may are interested in for a specific trial, there's a design. So what assumptions should we consider to determine the study size based on the design? For phase 2 trial, should we plan an interim analysis? So for operational lead, they could say, for each trial, based on the study size and the GL strategy, we will determine number of sites to be included to recruit patients. We may have some more experience in some countries, but not in all countries. And for regulatory lead, they would say, we need to get a CTA, clinical trial application, before we start each clinical trial, depending on where the patient will be recruited. Phase 1, phase 2 trials are exploratory trials. For phase 3 trials, we should strictly follow regular guidance to design the study, and overall alpha needs to be controlled. So this is just one, four stakeholders are listed in this example. Besides these stakeholders, there are many other stakeholders, including project management, CMC, represent chemistry, manufacture, and controls, commercial, clinical pharmacology, pharmacovigilance, and safety, and discovery. So all those stakeholders, statisticians will work together and interact each other during drug development. These slides are just show you another example, cartoon example, to show, I think, a biopharma statistician have a lot of interactions within data management, style programming, and system design. So here around the table, you can see we have a statistician, data manager, many handling data, and a SAS programmer, RT system build designer. So for a study, statisticians maybe say, oh, for this phase 2 trial, it's a standard parallel double-blind study. We need a RRT to randomize subjects and EDC to collect trial data. Data manager maybe say, in EDC, we will build automatic queries and audits checks to ensure quality of collected data. Clinical database will be CDC compliant. For RT builder, we need dummy randomization schedules for subjects and kits, and production schedules for subjects and kits from the trial statistician to facilitate the vendor to build the RT system for this trial. And the SAS programmer may say, oh, in this trial, we need a variable X, Y, Z collected to derive analysis variables for the analysis per SAP. Item data set will be created to support analysis. So this is just a simple example that the biopharma statistician could experience during drug development. So you can see from these two cartoon examples, we mentioned a couple of the many terms that are used in clinical trials, and biopharma statistician need to get familiar with. So in the following, I will introduce those basic concepts in clinical trials and terminology. So first important question is, what's the clinical trial? I think some others already know that clinical trial is a prospective intervention experiment to evaluate effect, example, efficacy and safety of an investigative therapy compared to a control. Control could be a placebo control, could be a standard of care control, or active control agent. So the protocol for a clinical trial will define how the trial will be conducted, including study objectives, patient selection, study endpoints, and how the data will be collected and analyzed. The statistical analysis plan, SAP, will define detailed data summary and analysis plan to answer study questions defined in the study objectives of the protocol. So the key elements in a clinical trial are the following. First one is a prospective design trial. Second one is include intervention treatment and compare with a control in a comparative trial. So the clinical trial conditions are well controlled and the study population are selected. So randomization for a controlled trial will ensure balance and a comparability of a baseline characteristics of a study population between intervention and the control groups. And lastly, blindness is to ensure trial integrity of data collection and endpoint assessment. So those highlighted in a bound are the key characteristics for a clinical trial. So clinical trials are usually classified into different stages. The trial design varies based on the trial objectives and the trial stages. So typically we will see the phase 1 study, phase 2 clinical trials, could be phase 2A, phase 2B, phase 3/phase 3B, and phase 4 clinical trials. Let's go to the phase 1 trial. Phase 1 trial is the first in-human trial after test the therapeutic agent, passes preclinical assessment, and works further evaluation in human beings. Study population in a phase 1 trial could be healthy volunteers or targeted patient population. Phase 1 studies could include SAD, single ascending dose study in patients or in healthy volunteers. MAD, multiple ascending dose study, usually is in patients. Typical objectives in a phase 1 trial include safety, tolerability, PK/PD, pharmacokinetics, pharmacodynamics studies. So sample size for a phase 1 study typically is small. For example, the phase 1 could be 8 subjects for active and a placebo with a 3:1 randomization ratio if you have a control group. So here I'll just provide 2 phase 1 examples as an illustration how the phase 1 trial looks like. The below example is just show is a parallel design with a placebo, 6 versus 2, 6 active and 2 placebo in a cohort for multiple ascending dose study with 5 cohorts. So each cohort could be is a 1 dose level. One cohort could be expanded to a larger size. For example, in this example, cohort 5 below is expanded to a larger size, but the randomization ratio remains at 3:1. The second example here is a medicine oncology trial. It's a single arm, is typically used in oncology area. I use a very basic example, the 3+3 design for maximum tolerated dose identification. This is very commonly used in oncology trial. So in the East, there is a Statistician software. They implement this 3+3. There are 3 variants of this 3+3 design: the 3+3L, 3+3H, and the 3+L modify. They implemented in the East software for the design. I just use one example on the very left. Is 3+L to illustrate to explain the procedure. So in this 3+L, the targeted probability of toxicity is 1/6. So when they start a trial, first they start a cohort of 3 subjects. So if 0 subjects experience dose-limited toxicity, defined in the protocol, they will escalate that dose to next dose level. If they observed 1 dose-limited toxicity, so they will stay at that dose level here and treat another 3 subjects. So total we'll have 6 subjects treated. So, but if they observe 2 dose-limited toxicity within 3 subjects, they will escalate the dose to the lower dose level. Suppose they treated a total of 6 subjects. They observed 1 dose-limited toxicity. They'll stop that dose and pick that dose as MTD, and so on. So this is the rules. Those are, I think, trialists use to do 3+3L. Similarly, there's a 3+H, focus on targeted probability of toxicity is 1/3. And another trial design 3+L modify targeted probability of toxicity is 1/6. So they have the all algorithm of how to escalate the dose or de-escalate dose. So in summary, here I'll provide a key difference of 3+ variants, the differences. If we observed 1 dose-limited toxicity out of 6 patients, 3+3L and 3+3H, we recommend dose escalation. But the 3+3L modifier will declare the current dose as MTD. If you observe the 2 DLT out of 6 patients, 3+3L and 3+L modifier, we recommend dose de-escalation. And the 3+3H will declare the current dose as an MTD. So this is just an example that's commonly used in the oncology area for phase 1 trial. So phase 1 trial could be a specific design for imaging, for example, MRI, PET, or others. And also could be a drug-drug interaction study, food effect study, TQT study, or other types of studies, depending on investigative agents, characteristics, and various regulatory requirements. In oncology clinical trials, there are some novel phase 1 designs. For example, Bowen's suite design, model-based dose finding design, CRM. CRM is a continued reassessment method design and a Bayesian version of those designs. So there's many designs. Those who conduct clinical trials in pharma industry, the statistician, they probably know more than me. So I'll just provide here some useful website for this novel design on the screen. But keep in mind, the FDA website is a very good website for the regulatory guidance for early clinical trials. So after phase 1, say if safety tolerability is okay, they move to phase 2 trials. Phase 2 trials may be proposed to test the safety efficacy for a treatment and using a relatively larger size. So the goal of phase 2 is to demonstrate proof of concept, POC. That's commonly used the term POC, proof of concept, or dose ranging for investigative therapy. Sample size per arm for a phase 2 trial is usually 60 or 80 per arm. Sometimes some trials have about 100 per arm for a double-blind trial, depending on trial effect size assumptions. Depending on study objectives, sample size stratification for phase 2 could be based on precision of estimation, just estimate, point estimate at confidence interval, or you could power this study based on assuming the larger treatment effect size for the two-sided alpha level is 10% or 20%. For example, for phase 2A, you already have a 2-arm trial with 1 active and 1 control to prove the concept. Phase 2B, we typically call it dose ranging trial to evaluate dose response for efficacy and safety to select phase 3 doses if the benefit risk is favorable to a new therapy. Phase 2B trial could include multiple active arms besides control arms. Could be 3-arm, 4-arm, 5-arm, or more. So in a phase 2B trial, you can do the dose response relationship in a phase 2B. It's very important to understand the dose response relationship for a new therapy. A phase 2 trial is usually designed as an exploratory trial, but it could be designed as a registration potential for some indications. What I mean by registration potential means you just like a phase 3, after you finish a phase 2 trial, you can submit for submission drug approval to regulatory agency. So this kind of indication is commonly happening for a rare disease. If we design a phase 2 is a registration potential, so you can support the regulatory approvals. So this was just to give you an example of phase 2B parallel design. This is a schematic. You will see in our study protocol. There's a schematic of the trial design. So in this example, the trial includes the following period. You screen a patient, a screening period. After the subjects are eligible, and you randomize subjects to different arms with 1:1:1:1 randomization ratio, equally randomized to one of those arm or placebo. Then they have a treatment period, they have visit schedules, and then after finish the last visit, in this example, is a week 24, you have a post-treatment follow-up period. Okay, after phase 2, If you prove the concept, drug seems to be working, typically we'll move to phase 3. So phase 3 trials are typically considered as the pivotal trials to support a new drug application. They call it NDA, new drug application in the United States, or MAA, marketing authorization applications in EU submission. So sample size for phase 3 trials is usually large enough to ensure a high probability of success. Phase 3 trials are typically powered with 90% power and a strong control with over type 1 error rate if multiple hypotheses are tested for primary and key secondary endpoints. Common assumptions for phase 3 sample size is power needed to be minimum 90%. Type 1 error rate is 0.025 one-sided. Proportion of missing data should be considered for the final sample size determination. Power is mainly based on the primary endpoint, but it could be based on a key secondary endpoints or combination of both. Regulatory agencies' feedback on the protocol and SAP for phase 3 trial is often obtained before start the trial conduct. In most cases, regulatory feedback are incorporated in the final protocol and SAP in phase 3 trials. So after phase 3, you get an approval. Then there's a typically some will have phase 4 trial. A phase 4 trial is usually a non-interventional study, and the primary objective is related to safety. Interventional studies could be planned to meet we call it post-marketing safety studies, PMSSS requirement. There are some specific terms for PMSSS. First one is post-marketing commitment, PMCS. This is from FDA. FDA may require PMCS for safety after a new drug is approved. There are two types of PMSSS studies. One is a volunteer PMSSS. Volunteer means a sponsor. They are volunteering to do this kind of study, but it's not required by regulatory authorities. The second one is a required PMSS. This is required by regulatory authorities. It is called a PASS, Post-Authorization Safety Study in EU, and a PMR, Post-Marketing Requirement in US. PMS, Post-Marketing Surveillance, they call it a PMS in Japan. So sample size for clinical trial, this is the main job for statistician to determine. It's a very important part. Phase 3 is typically power with 90% power with a strong control of overall type 1 error rate in a trial. If multiple endpoints are included for primary and key secondary endpoints. Common assumptions, as I mentioned earlier, for a phase 3 trial, power need to be minimal 90%. Type 1 error rate is 2.5% one-sided. Missing data need to be considered for final sample size determination. Phase 2 trial size depends on the trial design and the study objectives. Estimation of treatment effect or proof of concept are typically objective for phase 2. So sample size will be determined based on that objectives. But sometimes the phase 2 trials is designed to be a registrational worthy, meaning rigorous enough like a phase 3 trial, so that the potential submission could occur if data turn out to be very positive. This becomes more and more popular for rare disease indications and sometimes for breakthrough therapies that show very promising results in early trials. For phase 1 trial size is small to evaluate safety and tolerability of a therapy. The size depends on the trial design. So sample size for clinical trials is typically calculated using validated commercial computer software. So we commonly use a tool as one is an inquiry, another is a yeast, or others. Sample size calculation are most common for most common design. Usually in a clinical trial are available in these two softwares. Sample size calculation procedures and packages are also available in some statistical analysis software like SAS and R. For specific test statistics published in literature and the calculation is not available in standard software, statistical software, computer codes developed by authors could be used to determine the sample size for a trial. It's preferable that codes are validated for accuracy before using them in clinical trial design and analysis. Below, I have two slides to show a snapshot of inquiry and yeast for sample size calculation. When you go into industry, if you use software, you will get familiar very quickly. This is the inquiry snapshot. So in this software sample size calculation, people may be familiar. It can calculate sample size for comparison of means, proportions, survival endpoints, counts for one group, two groups, or multiple groups. So the yeast have a similar functionality. They have similar functionality just like inquiry. So yeast can call RR codes to perform simulation and analysis. Commonly used procedures for type 1 error rate control are implemented in yeast. Those procedures include Bartolini method, Huckelberg procedures, Holmes procedure, fallback procedure, parallel stereotyping procedure, and a graphical approach for overall alpha control. So another important topic for statisticians is overall type 1 error control in clinical trials. So typically for phase 1, phase 2 clinical trials, the overall type 1 error rate is typically not strongly controlled. For phase 2 trial, two-side 5% threshold or one side 2.5% threshold can be used for statistical testing for each endpoint. For phase 3 trials, it requires strong control of the overall type 1 error rate to be within two-sided 5% level or one-sided 2.5% level using closed testing procedures. There are many methods and procedures to control family-wise type 1 error rate. So I'll provide a link for a review paper. You can look at the details of those procedures. So commonly used methods include Bartolini method, weighted Bartolini method, Holmes procedure, Huckelberg procedure, fallback procedure, fixed sequence procedure, Holmes procedure. This is for single stage overall alpha control. For parallel and serial keeping procedures or graphical procedures can be used for overall type 1 error control for multiple families with multiple endpoints. Group sequential procedures can be combined with alpha control procedures to control overall type 1 error rate if interim analyses are planned in a trial. So currently, yeast software implemented most procedures for testing mean difference or proportion difference or mix of these two type of endpoints using complex alpha control procedures. So here, I'll just give you an example of a graphic approach to control the overall type 1 error rate for multiple doses and multiple endpoints. So in this clinical trial, we have there's primary endpoint family, but we have 3 doses, high dose, mid dose, and lower dose. And there are 2 secondary endpoints. These are 2 secondary endpoints with 3 dose levels. This is another endpoint for 3 dose levels. So you can see this is a typical graphical approach. At the very beginning, you reallocated alpha, say 5% alpha in the primary endpoint of different dose levels. And then you define how this alpha will propagate among different endpoints and across different points so that you can use all alpha after one hypothesis is past statistical significance at that level. So I think if you get an industry you are interested in, you will see if there's a complex design. So this graphic approach is becoming very popular in industry. So in early cartoon examples, we also mentioned about several terminologies. So there are some abbreviations biopharma statistics often used in our daily work life. For example, EDC, electronic data capture, RRT, iterative response technology, TSUR is development safety update report, PSUR, periodic safety update report, SAP, statistical analysis plan, DMC, data monitoring committee, TMF, trial master file, IA interim analysis, DBL, database log, CRF, the case report form, CDIX, clinical data interchange standards consortium, defines standards to deal with the medical research data linked with life science. SDTM, study data tabulation model, create and map, collect data from raw data source, and analysis animal model, derived data set that are ready for analysis, i.e. analysis ready data set, animal. So here I just go to more details about each terminology. EDC. So EDC is electron data capture interface. On the left, just is a snapshot. You can collect demographics. And of course, the EDC can collect more in clinical trial data. So they can collect the patient demographics, medical history, medication use, clinical safety, AEs, trial inclusion, exclusion criteria, trial visits, lab, vital sign, weight, clinical efficacy assessment. In the EDC, we typically build queries and logic audit checks in a system to ensure data quality for data collection. EDC system specification define variables for trial to be collected. Data collected by EDC is usually reflected in the clinical database in a timely manner. A lot of time is a daily refresh. RRT, that's another important system that statisticians are involved, is an interactive response technology. It's a computer system to conduct randomization and drug supply management for a trial. It helps sponsors at the investigative sites to manage subjects and drug supply logistics in clinical trial. It can be integrated with other clinical trial systems. On the right, I just draw a diagram to show you how the RT work. So RT in the middle here, they have interaction with reporting and analysis, subject randomization, drug dispensing system, and drug supply management system. There's a web interface to site, sponsor, and other parties who need RT. And also they could be integrated with EDC or other clinical systems. So statistician major part involved is randomization. Randomization includes subject randomization or kit randomization. These two schedules for the RT is the main contribution from trial statistician. Schedules generation, system testing, we call the UAT, user acceptance test, for randomization shall be performed before the system goes live. Other functions are involved in UAT include drug supply, trial operation, clinical, data science. So DSUR and PSUR we mentioned earlier. The DSUR Development Safety Update Report is a periodic reporting on drugs under development. So if you have trial ongoing, that means the drug is under development. So this includes marketing drugs that are still under development among the ICH regions. Report needs to be updated annually. So the focus of DSUR is data and findings from investigative clinical trials of drugs and biologics that are under investigation, whether they have been approved or on the market or not. PSUR, Periodic Safety Update Report, this is a pharmacovigilance document intended to provide evaluation of risk, benefit or balance of a medicinal product at a defined time points after its authorization. Safety for approved drugs. The contents of DSUR, PSUR could be overlap, information for market experience. The reporting of PSUR might be relevant in clinical development and should be included in the DSUR as well. Both DSUR and PSUR should be comprehensive and stand alone. So this is another terminology, SAP, DMC, and TMF. SAP, the statistical analysis plan. This is a document, the author is statistician. They document detailed interim or final analysis or data collected in the protocol after database lock. Analysis should include subject disposition, summary, baseline demographics and characteristics, medical history, and concurrent medication summary, analysis efficacy endpoint, sample size, table error control strategy, safety analysis, table listing figures contents for clinical trial report, CSR. So DMC, they monitor safety or efficacy data at the interim in a trial, could be internal, meaning sponsored personnel participating in DMC as a DMC members, or external or independent DMC. In this case, all DMC members are from outside of the sponsor that have no conflict of interest for a trial to be monitored. A DMC usually include a chairperson, physicians, and a statistician. Experts from other fields could be included in the DMC based on the scope of DMC activities and responsibilities. DMC activities are governed by a document called DMC Charter. The charter could be prepared by a statistician or medical director, depends on the process defined in a company SOP. Because different companies have different SOPs, standard operational process, so the author could be different. Last one is the TMF. It's documented all trial activities for traceability and ready for potential future regulatory inspections. More terms, i.e. the interim analysis, the planned or unplanned statistical analysis of the interim data before the final data is locked. Could be data review on safety or efficacy or both. So interim analysis is a statistical analysis of interim data to support a go, no-go decision-making, including interim analysis for futility, early stop of efficacy, acceleration of development, sample size re-estimation, adaptive design, etc. So interim analysis could be planned in any phase of a trial. If planned in a phase 3 trial, in a registration trial, overall top-line error control due to multiple analysis must be considered for efficacy analysis in SAP. DBL, database lock. This occurs when all protocol-defined procedures are completed and the protocol-defined data has been collected and cleaned based on the trial data management plan. Different companies have different procedures for database lock. Once database is locked, log data shall not be changed for the final analysis. If changes must be made for locked database, specific documentation should be in place to explain the rationale for unlock and relock of the database. Lastly, CRF, eCRF is case report form or electronic CRF. This is the fundamental tools for data capture in clinical trials. Serves are guidance for EDC to build and fundamental documents for statistical programmer, statistician to prepare analysis derivation and analysis plan. Okay, next is a major topic is clinical trial reporting. There's 3 major terminology: CDIX, STTM, and IDO. And lastly, is table listing figures. CDIX standard is a clinical data exchange standard consortium, which is defined standards to deal with medical research data linked with life science. So the CDIX is a disease specific. So on the right, I just include a snapshot of some CDIX specific for disease. You can see that acute kidney, Alzheimer's, asthma, CIPD, there's many diseases, COVID-19. So different diseases, they have their own standard. So if you do a clinical trial, maybe you can go to our website to learn more about it. STTM standard. So STTM provides a standard for organizing and formatting data to streamline process for collection, management, analysis, and reporting. Implemented STTM supports data aggregation, and warehousing, boosting mining and reuse of facility sharing, help perform due diligence and other important data review activities. So SDTM is used to create and map, collect data from raw source. ADAM is all about creating data that is ready for analysis. So SDTM is always the source of ADAM data. It could be a domain or a supplemental qualifier, but a source is always the SDTM datasets. So SDTM is one of the required standards for data submission to FDA and the PMDA in Japan. So I have provided a link for FDA and the PMDA guidance on standardized data requirement. So ADAM standards. So ADAM define dataset and meta standards to support efficient generation, replication, and review of clinical trial data analysis. Traceability between analysis results, analysis data, data represented SDTM. So ADAM is required by FDA and the PMDA for new drug applications. So the other required sources FDA and the PM website. You can go to that link to learn more details about ADAM. So table listing figures. This is the product of a clinical trial. So after you finish the clinical trial, you will generate tables listing figures to give the physician or different stakeholders to review. So commonly, this table is including subject disposition, early discontinuation of study or study drug, medical history and concurrent medications, baseline demographics, disease characteristics, efficacy, safety. Safety is a major part, very common across different indications. So overview of adverse event, AE summary by preferred term or system organ class, lab change from baseline tables, hematology, chemistry, and urine labs, and the lab shape tables. And also, safety could be a potentially clinical significant event or lab values. We call it PCS. And vital signs and PCS for vital signs, ECG, and CSSRS is a Columbia Society Severity Rating. This is very common for neuroscience clinical trials. And the listings. So let's just typically display the raw data of a clinical database. Okay, I finished all the selected terms interpretation. Here, I just would like to give a final comment, remarks. So daily work life as a statistician in biopharma industry is busy, interesting, and meaningful. Statistical experience makes difference to drug development and eventually a benefit to patients. Many interesting and challenging statistical topics observed in drug development process need solutions from statisticians. Biopharma industry is a highly regulated industry. Statisticians in biopharma industry not only need operational excellence in clinical trials conduct, but also are good at scientific excellence to address challenging questions in the daily work life. Rewards are fruitful and achievement is touchable. I mean here, achievement is touchable. You can feel your contribution to the patient. But the challenges are sometimes is big. So in a biopharmaceutical company, there are various statistical expertise are required in different therapeutic areas and different clinical development stages. In a large biopharma company like AbbVie I'm working for, this is my employer, statistics groups could be organized into different spatial groups based on expertise. In our company, we have clinical statistics for early stage or late stage, design and trial operation, as non-clinical statistics for CMC, animal studies, exploratory statistics for data mining and biomarker analysis, innovative statistics, handling statistical methodology research for clinical trials and application in clinical trials. Safety statistics focus on safety analysis and reporting, etc. Statisticians are not working alone in biopharma company. Collaboration with team members from different functions are crucial for his or her successful career development in a company. So I would emphasize the collaboration. It's very important. It's a key. So I think that's all I have today. And thank you for your attention. And we can go to the Q&A stage.
Yeah, so thank you, Deli, for such an amazing presentation. I'm not sure how you really like explain so many things in this such a short time, but I'm sure like everyone like me, maybe we already have a very grasp of what's really like to be a statistician in pharmaceutical company. But of course, I feel like maybe some of our audience feel also overwhelmed with all the terms. So I already see some question about the slides. So we are going to share the slides and also the recording for this webinar in the follow-up email. It should be sent out like in 3 to 5 days after this. And you can see also that Deli actually provided many links, helpful resources. So if you would like to dig into those resources, you can just follow the slides. We do have 5 more minutes for questions. I think we have at least 2 questions in our chat. So the first question is from Hao Yuzhou. So the question is, can you please elaborate a bit on the selection of the postulated effect size in the calculation of sample size? Maybe an example for the sample size calculation in phase 3. Really, I'm not sure if you can see the question in the Q&A.
I didn't see the question. Can you repeat? I'm sorry.
Yeah. Can you please elaborate a bit on the selection of the postulated effect size in the calculation of sample size? Actually, I'm not really sure about what's postulated effect size.
Yeah, postulate. Okay. So I think it probably is assume the sample effect size. I think in phase 3, this is a very good question actually. For statistician, should really care about the assumptions. Typically, we work up with the clinical team to find the assumptions for the sample size determination. That assumption is the major one is the effect size. For example, if you use a continuous variable, typically you have mean, for example, is the mean difference. You have placebo treated, what's the mean difference of that? And what's the common standard deviation? So you can calculate the coherence effect size, the mean difference divided by common standard deviation, and you plug in the yeast and or inquiry to calculate sample size. But I think before you calculation is very straightforward, but how you get that effect size is very important. I think there's many ways you can do it. One way is that suppose you are in a trial, there are many studies already published in the literature. So you can collect those data together and then you can do meta-analysis. Okay, you can estimate the effect size and use that for calculation. Or secondly, if your competitors have similar trial and they will publish results, or you go to a conference, they present, and you get those information and maybe as a reference for your trial design. Depends on the compound and how good the treatment effect is. Typically, clinicians have more sense regarding the treatment effect. It's kind of like when we determine effect size, you have to work with the clinical team to define a final effect size and align within the team before you calculate. Of course, sometimes you can do simulation. You try different effect size and current sample size, or vice versa, and you can determine where the sample size is because effect size is related to sample size and power. So that's how for the typical situation. But for those trials that there's no published results, and then I think probably likely you have to do some assumption. You assume I want we call it TPP, targeted profile. So our commercial, our drug won't deliver, say, 20% slowing or improvement of drug compared to placebo. So based on that, and then you can come out, okay, the treatment effect size, and you can calculate the sample size. So this scenario, I think there's a lot of assumptions. Part of another day, we do clinical trials, we cannot work on without any data. So that's why the clinical trial have different stages. Okay, phase 1 mainly for safety. Phase 2 typically can generate some efficacy data. Based on phase 2 results, you can get the effect size estimate, and then for phase 3, you can do the sample size calculation. I think that's the phase 2 is required typically, but sometimes some drug is breakthrough therapy. That's huge treatment effect, and then for statistician, it works very easy. Okay, so effect size, for example, is 1. Then you need 10 patients is enough. It depends on situation. So there's many situations. I think we have to work together with clinical team to come out effect size and help statistics to determine the sample size. So I hope I answer most questions.
Yeah, thank you, Deli. I think we may have time for answering another 1 or 2 questions. So the next question in the chat box is, can you share more about the development about SAP, statistical analysis plan? How do professionals collaborate in that?
Okay, sure. SAP, the main author, the statistician is also main author for the statistical SAP. Typically, we are doing so you have a protocol. Protocol is, within the protocol is a statistics section that's also drafted by a statistician. Protocol will work together with physicians and all others to write the protocol and follow the protocol based on the data collection. And we define analysis. For analysis, in different companies have different template, but the major components that you have, a definition, derivation, data set, which data you're going to analyze, ITT or for analysis set, safety analysis set, and then for efficacy, I think will be depends on indication and trial, they have different requirement. But specifically, I think there's an FDA requirement or EMA requirement. For specific type of data, you need to use a specific analysis tool, especially for phase 3. But for phase 2 or early clinical trials, you can use whatever methodology available, statistical methodology available. You can analyze it, no problem. But for phase 3, for overall type of error control, because FDA and the EMA, they have a strong requirement. Because some methodologies we all know, they call it we all know methodology, the strong control of alpha, you can use it. But if you propose a different methodology to analyze the data, you have to convince the regulatory agency, okay, this methodology is sound. There's some publication support in the literature and also strong control over alpha. So that's typically how we handle it. But we usually follow FDA's guidance or regulatory guidance for analysis methodology. But for some cases, some methodology is not sensitive. We propose a novel methodology is sensitive, but we have to prove methodology-wise strong control over alpha.
Yeah, thank you, Deli. Maybe we can try to address one more question. Might I ask if you can give some example of non-standardized clinical trial?
Yeah, I can give you an example. Currently, I cannot name I was involved. We could call it non-standard, but I don't know how the non-standard. For clinical trial is typically with placebo-controlled trials. Sometimes we have used single arm trials with external control. For example, for some rare disease, I just tell you there's one indication I was involved called Weiner disease. So in the world, it's just like total is 300 patients alive. It's a very bad disease. It's hard to find a patient. In that case, we cannot do the double-blind control trial. We cannot do it. It's not feasible. In this case, you have to think smart. How can we do a trial to show the efficacy, the drug work for this population? That's all I can think of is one example in my head right now. Of course, there are some other examples. Maybe I never experienced, but there are some non-standard clinical trials. In a later video, if I have some, I can never share. That's all I can think of. So in my experience, that's one I can think of. But I think I heard probably in oncology trial, there's a lot of maybe non-standard, but I'm not aware of that.
Okay.
Thank you.
Yeah, thank you. I think we are running out of time, but at last, I would like to have some acknowledgement to our ICSA committee who really made this webinar series possible. So these are all the members from ICSA webinar subcommittee, and some of them are going to serve as speakers for the future webinars or facilitator or panelists. And as I mentioned that we are launching different series of webinars, and we're going to provide the registration link in our follow-up email for all the future webinars. And also we are going to provide the links to the past webinar recordings if you are interested to explore and if you missed it earlier. And so at the end of the webinar, after you leave the webinar, it will pop up some survey. So if you have any suggestions for future topics or any suggestion for our the format of the contents of the webinar series, please just let us know. And thank you again all for joining us today. I hope everyone have a good rest of the weekend. Bye.
Thank you.
Thank you.